Okay, so thank you very much. As you hear from my job title, I'm now a full-time pen pusher, but until recently I was still a scientist. So I'm going to talk about the work at the Rosalind Franklin, which is just across the road from Diamond, for those of you who haven't seen it. So this is the building, and this is the work of many hands, always most importantly, to make sure I call out all the people. So I'll try to talk about the work as I go past. But there's a collaboration actually between Diamond, Thermo Fisher, LMB, and the Rosalind Franklin. It was funded by the Wellcome Trust. It's kind of a 10-year ambition to be able to get into tissue from patients ultimately and start looking at structures inside chunks of tissue rather than in model systems. So for those of you who know this, young Richard Henderson, busy electrifying life science over here on the right. It actually says Soviet power is electrification plus hard work or something, I think. So the idea is to get out of the lab into the human. I think that we kind of feel we're approaching the limits of complexity that we can get at in isolated cells. So if we really want to get into understanding disease, we are going to have to look at tissues. So what we do today in proteins, and that was a motto in the Franklin, is that we will do in cells and tissue. So not just structural biology, but chemistry, mass spectrometry. The whole drive at the Franklin was to get out of model systems over a 10-year period and get into tissue. And so we can actually bring our technologies to bear much closer to the clinical need. And we're doing this at a time where the genomics revolution has come and to some extent gone. It's going to be the revolution. Next one is AI, which is going to allow us, I hope, to merge data streams in a way that human brain doesn't do very well. And you see, I think, the beautiful work of Pei-Jun, and I'll touch on this idea that we have so much data of biology from mass spectrometry, from other things, that really, if you know your protein, that's probably, you're doing pretty well. But you don't know much about all the stuff that's one step removed. But the great thing about AI and machine learning is it can integrate all those different streams of data. So we have connectomics, we have proteomics, we have cell content analysis. All of that stuff is kind of known, but it's very hard to feed that into structural interpretation, because you know your thing and don't know the others. So I think AI is going to make a huge, huge play here. Once they stop making tons of money from telling you what to look at in Amazon, which is mostly what they spend their time doing. So why is structural biology particularly important to this? The function always comes from the farm. So my background was in enzymology, and of course so how I've always worked, knowing the enzyme, you know the function. But there's no better way to understand biology than seeing the atoms. And structural biology has been really good at curing the diseases we face. HIV that Pei-Jun talked about is a great poster child of structural biology. The protease inhibitors, for example, have made that disease, while still serious, no longer a death sentence for so many people. But really what we will get at, I think, with this approach is we'll see disease at a much earlier stage. So we don't really know the molecular events behind many chronic diseases like inflammation. We don't understand the earliest molecular events in Alzheimer's and in other degenerative diseases. And often viruses will do other things inside the host. You get chronic responses like long COVID. We don't really understand what the molecular drivers of those things are. So looking inside the tissue is probably my best bet for how you will get there. So this is a standard slide that most of you have seen, but the idea is that what we really need for biology there's no one technique will get us from atoms to tissue, but it is stitching together these different data streams that will allow us to actually get this dream of being able to look inside tissue all the way down to atoms. So a little bit of the puzzle that Franklin Diamond, in fact, work on. So we collaborate with Diamond around this area. Okay, so this is a focus for the Franklin. The Franklin focus is down in this regime. With Diamond and Oxford, the focus is up in this regime. Particularly the new hard X-ray micro-CT lines, I think, look really interesting. So tomography, electron tomography has a long history. There's no better exponent than you heard by Pei Jun. You go back to the early papers here with lift out and other approaches have really begun to change the field and show what can be done with it. My own view is that the other revolution, which is just as important here, has been the ability to get de novo structures. Of course, it's really a cheat. They're not de novo. It's able to recognize patterns. But nonetheless, from a practical point of view, de novo structure determination of protein fold is no longer a respectable career. Right. I always say, no doubt somebody will stand up and then tell me you're wrong. We should celebrate that I can't imagine asking a student to determine the structure of a protein now. That we would simply take the AlphaFold model and start doing the functional work that we really claimed for years we were most interested in, do that. And if that gives contradictory results that we can't interpret, then and only then it might be worth going back to look at the structure. And the law of computation means this is getting better, faster. So I think this is going to, be 99% done in just a few more years. And we'll soon unlock complexes, certainly binary complexes, ligand complexes, all of that stuff will fall to this approach, I'm absolutely certain. And we should celebrate that. That means it brings within our reach to be able to put molecules inside cells. We can't determine de novo the structures inside cells very easily. So you saw the beautiful work of Pei Jun, but that's very hard work. And if you've got hundreds of different proteins in the cell, what you really want to be able to do is just recognize them roughly and then fit atomic models from AI inside that, is my own view. And I think that now is the time. So the tomography in a box concept is one of the things that Franklin was set up to do. So tomography is where all the structural biology, I think, will go. In electron microscopy, we will move out of single particle in a relatively short period. But we'll definitely get into the cell. And so the idea of the Franklin was to make this simple. So we have a workflow that we call tomography in a box, and it has this components: think, locate, shape, measure, and see. Now, I'll only briefly talk about think and see, but they relate to this AI question, the ability to recognize people or three-dimensional objects and then pop in pretty good AlphaFold models inside them. So in thinking, what we do is we've created, and this is a program called Parakeet, which is freely available, so I won't talk too much about it. But it's an ongoing development project where we're simulating large volumes containing proteins and simulating tomograms, adding noise. We've just started to include particle-induced motion at simplistic level. Still some way away from being perfect, but the goal here is to simulate tomograms of collections of proteins well enough that then you can use that to start training AI to recognize three-dimensional shapes. It's a three-dimensional shape recognition problem. Now, AI is pretty good at these things. That's why your phone unlocks when you look at it. It's a facial recognition problem. And facial recognition is two-dimensional, but three-dimensional problems are more complex, but not beyond the bounds of possibility. So on this side, it's about trying to create high-quality, realistic training data at scale, but it's also, for us, about designing the experiment and the workflow, trying to understand what factors really influence data quality. And then we'll come back to that when we get to measure. So locate and shape. So how do you find the bit you're interested in? So, and I'll also show you some work in fluorescence, and Pei Jun showed you how powerful that is. But when we get into human tissue, we are not naturally fluorescent. And it's unlikely that we would get permission to genetically engineer the population to stick in GFP into the target region. So we need some other way to find the region of interest, and we'll talk about that. So the thing that the Franklin did, the first thing it did was work with Thermo Fisher to get these cryo-prefibs running, and there are two of them in the Rosalind Franklin. So cryo-prefibs are nothing special. They're just essentially very similar to the gallium prefib, except at high current. So the unique advantage of plasma fibs is that you can work at a higher current. There's nothing, and I'll show you the data that shows they're no more or less damaging at least for biological samples, than you can meaningfully measure than gallium. But the point is you can go at much higher rates, especially for xenon. And that's important if you're starting to take chunks of tissue. So the first bit of work then is taking, this is from Michael Grange, who's a young investigator and welcome fellow at the Rosalind Franklin. So here you have mouse brain. So this is brain from a mouse frozen in the lab in the morning, high pressure frozen. And then what we're doing is we're PFIB milling it. In this case, it's an argon beam, and you're stripping off the surface layers. So essentially you reconstitute. It's not a true tomogram. It's essentially 2D projections. And each projection is about 50 nanometers, 30 nanometers, depends on the electron depth. But it simulates, looks like a volume as you slice through it, and you can see, look here, we have the synaptic junction inside. So this is from a real live mouse brain. Well, it's not live anymore, of course, but you can get extraordinary detail in tissue. Now, the importance of this is using an L-star column that we have in one of our PFIBs. And the performance of L-star is really good. And actually, I think it's probably a mistake in the new generation of the PFIBs that Thermo Fisher are selling, is they've gone back to a Nicolay column, which is not as useful. This L-star column is going to be critical for the tissue work that we do, and I'll show you why. Because you can correlate images directly with SEM and TEM now. So this is just another example looking at mitochondria. So this is from a HeLa cell. You see it runs through. You can see all sorts of nice stuff. So these are SEM images, and with L-star column, you can actually segment out whole chunks of structure. So you can see here, we're pulling out the centriole. So you can actually do structural biology. We were able to see the nuclear pore complex. Actually, just go back. You should be able to see it as we go. You see the nuclear pore complex appear just here. And this is SEM imaging. This isn't TEM imaging. Right? This is unstained native SEM. So I think that what I'm going to leave you when we get to the correlation is that with SEM and fluorescence is useful for today's problems, but it won't be useful when we get to tissue. But we can correlate fluorescence, SEM, and TEM. Right? So this is the same lamella in SEM and then done in TEM. So this ability for us to be able to look at tissue as we slice through it, say stop, take that region of interest, but correlate our TEM image into that larger volume, I think is going to be really important as we move into whole tissues. So one of the questions that we faced when we and others started to use plasma milling was, it will be much more damaging. And it turns out there's not really very good literature on how damaging even gallium fibs are. The best data probably came from Dave Stewart on when they used it to polish crystals. But there's very little on looking at surfaces, what actually is the damage depth. So there's more work now, but I think we set a way of doing this that's really quite nice, and that's published in Nature Comms. What you're doing is segmenting your ribosomes in a sample by depth from the surface. So essentially, you find all your ribosomes in your lamella, and then you reconstitute your subtomographic averaging by depth, and you look at the structure, essentially, of all the ribosomes that are close to 0 to 15 nanometers, 15 to 30, all the way down, okay? And from that, then, you look at the structures. You're able to get very high resolution structures, as high as NMD is actually slightly higher than had previously been done. This is from E. coli. And the bottom line is that the damage ceases about 40 to 50 angstroms, 40 to 50 nanometers. So, you're done. There's no real damage beyond that. Okay. So it's very little damage around. You can't measure meaningfully damage above 45 nanometers in the sample. And of course, in the 0 to 15 region, you're actually cutting into the ribosome itself. So the damage will in part reflect that there'll be partial ribosome structures in there. Others have then followed up with gallium and come with a relatively similar number. In a paper that's now under revision at Nature Comms, which is kind of bad luck to say that because you never know. The revision should go in this year, in the next couple of months. But what we've done now is use very high current xenon. So xenon can ablate right through your sample. So you can work at 200 micrometer thick samples and ablate with xenon. And we're able to show that as long as you slow down as you get close to the region of interest, you can get similar results to the very polishing argon beam, but we didn't have to polish out the damage here. So xenon gives very good structures. Again, we're below 4 angstroms for whole bits of the ribosome. The cutoff is undetectable by the time you get to 35 angstroms, but you can certainly live with 20 to 30. So we're able to go at pace now, and that's one of the things that was really key, was being able to take large volumes and whittle them down using xenon at speed. So it really brings within scope tissue plugs. So of course, this is all highly manual at the moment, and so we've been teaching a machine to look at lamella and try to see if it can mill much more automatically. Because what we found is the automatic milling recipes aren't very useful for tissue. Tissue has all sorts of quirks in it, so bits mill faster than others. So what you really want is some dynamic feedback looking at the image. And what we're using is the GIST layer. So again, for those of you who don't know, to stabilize a biological sample before you do plasma milling, you will coat it with a layer of platinum. And you can actually image that platinum. And what you can use is, because of the geometry of the way the lamella are cut, then you can use the size of this clearly defined GIST layer to tell you what the thickness of the actual lamella is. So you can try to dynamically get that to feed so that you just say, I want a lamella this thickness, go. That's the goal. So measuring the sample, of course, is a TEM image, and this is taken from Michael's lab. And so Michael is measuring bits of brain directly and looking for early molecular signatures now inside brain tissue for Alzheimer's disease. So in the last, I think, 5 minutes, I'll tell you a little bit of work that I have been doing with a student in my lab who will be my last PhD student. But also, as ever, I've been very fortunate to have some of the best. So TIR domains are really common in biology. They're found in all sorts of roles, but they seem always to aggregate. So they always seem to stick things together. So you turn up in both eukaryotic, prokaryotic biology, and this TIR domain always seems to be in some way making things in assembly. Now our interest is in a collaboration with my long-standing collaborator, Malcolm White. So Malcolm is very interested in antiviral defense systems, and of course this is what won Jennifer Doudna and others the Nobel Prize. This is where Cas-CRISPR comes from in this collection of response against infection. But Malcolm's particular interest is in here. So these are the CBAS system, but the bottom line is when the cell detects that it's been infected, so it looks for cyclic oligonucleotide production, it then switches on an activator and kills the cell. Now Malcolm's lab, along with Laura Spagnola, had shown, so here is your TIR-SAVED protein. So this is a TIR-SAVED system, and it's a CBAS effector. What you know is that in the presence of this trinucleotide here, so this is this trinucleotide adenosine, it binds to the saved domain, and that then undergoes a conformational change, which leads it to form these large, these were seen in single particle, these slinky style springs inside on an graphene grid. But what Malcolm has shown is that there's something really quirky about this, in that the reason you need at least 2 molecules is the active site is partly contained on one face and the other face. So when you make a dimer, you complete the active site. So you can understand why you need a dimer. But what Malcolm has shown is the dimer in and of itself is not effective. So the dimer is not catalytically active. It requires some form of higher assembly inside the cell than a simple dimer. So there are tricks to make dimers, right? But they are not active. So you need this higher assembly. So with Helena from my lab, what we did was we found a way to grow the cells, E. coli, and then we use a mutant that is inactive because otherwise initially when we did this it would kill the cell. And then we have a system that generates the tri-cyclic nucleotide. So you've got two systems you have to induce. But then in E. coli, you'll see when the movie clicks back, you see here, and I'll show you in a bit better, but we suddenly could see these fibril slinkies growing inside the E. coli. Okay, so we're able to see them inside the cells directly. So that was nice because previously it had only relied on it being in vitro as to whether this was really real. And one of the things that in the paper had been critiqued was that, well, this was all very well, but did these fibrils form inside actual living cells? So here's an example of a tomogram. You can see here we have the bundled side view of the filaments. We also have individual filaments. We can see down the top view, and we can see side views of the filaments here. So of course, here's a ribosome, and you can see these large bundles. So we're able to see inside the cell, and we can see the bundles and the filaments. When we look at the hexagon, it's a beautiful hexagonal close pack. It's almost perfect. You see here, there's only very slight metric distances between these bundles of helices. We have been able to improve the resolution now, so this is the latest resolution. And we now detect that we have two pitches of these slinkies inside the structure. So one pitch has a twist of 22.6 and a rise of 8.1, and the other has a shorter one. And you see they don't superimpose, but what is more intriguing to us anyway from a biological viewpoint is you can see inside one of them, and I'm going to morph, there's something in the middle now. And you can see as it grasps whatever it is, it tightens the helical pitch. So whatever this is, and we don't know what this mystery molecule is, it results in this breathing motion. Now, we are at the resolution now where we should be able to put the monomers back into this structure. And that will be important because that will tell us whether or not on how these filament bundles interact with one another. Helena has also begun to actually use the native protein, and here the game is to try to freeze frame before the cells explode and die, the beginnings of the what's actually happening in the cell as you activate this system. What do you actually see? So this is just to show you, this is this thing in the middle. It's intriguing. It pops up quite often, but it's not the full length of the helix. It has a defined length within a fibril. So we can slice through a single fibril and we can see it starts and stops. So it's not just a continuous string of density. It does slice along. So we have measurements to do to get some measurements in that. Now I'll finish with the last thing that Franklin will do in its 10 years of electrifying life science. And this will be the new TEM machine that will arrive towards the end of 25. And it's called Hodgkin. And so it will test for the community a number of things that we all believe are really going to be really important. So this is taken from a paper, I think, from Chris Rousseau and Richard Henderson. And what they show is that if you are able to chromatically correct, in other words, rescue inelastic event electrons and focus them, you should be able to shift at 300 keV, the resolution. So you should be able to dramatically improve the resolution of our data. That's the bottom line. So at a 600 nanometer thick sample, right, you should be able to get much more information than we've done before. So we should be able to move to thicker samples. So our target is to get to 500 nanometer thick samples because that's a much bigger chunk of the cell, much more relevant to the tissue samples we want to do. So chromatic correction is key to that. The second bit of technology the microscope will have, this is a chromatic corrector. It means the building, which house it's going to be very high. It has a liquid helium stage, which will allow us to, but it's not just one temperature, it's a controllable temperature. And Richard and Chris have shown that as you approach liquid helium temperature, you do increase radiation hardness. Now there's a point where you don't want to go all the way to helium, otherwise you trap hydrogen. because it doesn't off-gas, but being able to probe systematically temperatures between liquid helium and liquid nitrogen and measure whether or not we do see any gain in liquid and radiation hardness, I think is really important for the community. And the gain could be significant. Right? We could shift the resolution significantly. So the combination of chromatic correction, liquid helium, and then the final technology we don't have the funding for, but we have the microscope designed to cope with, will be the laser faceplate. Chromatic correction works best when you are at the focal point. So does the laser faceplate. So these seem made in heaven as a match. So the microscope will test the combination of chromatic correction and laser faceplate to maximize the information content. We'll be able to work at focus on thicker samples. So the goal for the Franklin then is, as you see, is to be able to derive the technologies that allow us to work on native tissue from humans and animal models directly without staining, without embedding, be able to put that back in context, but be able to take these highest possible resolution tomographic images with the next generation of faceplate, chromatic corrector, and we'll see if we can put more electrons in, which always helps. So with that, thank you very much.
Hi, fantastic talk. So I have a question about the fibrils you described from your student. Why is it that you need a higher order oligomer to get it?
That is the million-dollar question. So it's not clear. Because the prediction was that dimers would be enough, right? And then it was, and it's not true. So it has to be some, either the thing is catalytically active as a dimer, but not as catalytically active, but you would still think it would be fatal because it chews up all the nicotinamide in the system. So that suggests that the fatality is not just the nicotinamide, right? But it may be these large bundles themselves some effect. So that's why the next stage is to start freezing cells as they're sort of dying in essence of the trigger. But the fibrils are essential. Thank you.
Very curious about the molecule that you trapped in the filament. Any guesses on what it could be?
Well, all my guesses are wrong. So, no. I mean, we've tried some things. We know what it's not. It doesn't appear to have the contrast of nucleotides, so we can rule out that it appears to be some sort of DNA or molecule, because originally we thought that's probably what it was with some filament, but it doesn't appear, because you saw in page runs that you get much darker contrast when you've got phosphorus. So it doesn't appear to be phosphorus. So ribosomes are much darker because you've got so it doesn't appear to be a nucleotide, because originally that's what we thought, but it's not that. The dimensions are hard because we're averaging round it, of course. So subtomographic averaging is designed for the filament, not for the thing in the middle, as we're currently reconstructing it. So we might be losing information about it. All we know is that it has a defined length, or appears to have a defined length.
Very nice talk. So with the damage layers on either side, I mean, it seems like an obvious thing to get to high resolution with our existing technology to try to reduce those. Are there kind of serious efforts going on to do that?
So my own view is that there's no need, right? The damage is trivial by the time you get beyond 35 nanometers. And in the first 15, you can't avoid that, at least with ribosomes, because you're knocking the tops off them. So your damage layer is still, you can still get a ribosome structure even in that initial damage layer. So you might be able to polish it out a bit, and we're trying that with low currents of argon. So one of the things is you think, oh, it should be easy to just split between xenon and argon. In principle, that's true, but actually it's not true in the hardware yet. So that's one of the things that we're trying to script, is so that, because you have to realign the machine when you switch. So that takes too long. But ideally, you would blast away with xenon, switch to argon, then switch to very low kV, and take, the last 15 nanometers out at that level. And that should get it down to essentially nothing.
Well, Jim, where do you see this in 10 years' time? You think this is going to be implemented in a way that can be going to many labs and eventually to, high complexity hospitals or things like that?
That's my view. Is I think the way it will work will be the SEM will go to labs, and there'll be a few of these monstrosities if they really work. So our goal was always, the Franklin was set up to do high risk stuff that would not normally be grant funded. That was his model. And so this was a challenge to test these technologies and see how robust they are. If they work, others will no doubt get them. Probably here will be one of the first. But they will not necessarily be limited in number because these machines will be 20 to 25 million, I think, when they get sold. And you'll need a building which is 2 stories high with stainless steel rebar and all sorts of complexity. So I think they will be difficult for places. So I would imagine they will cluster in national centres. But what you will be able to do is use the PFIB recipes, the correlations that we're developing now, so that you whack out your samples, they go in the dewar and they go off to these things.
So let's thank Jim again.